Hi, Michelle Glass here. Our last little bit of conversation here on Chapter 6, Lecture 6, is to look at that relationship between food and ATP a little bit more closely. So we've been talking this whole time about how the cell will break down glucose in order to make ATP. And we know that glucose is a monosaccharide or monomer of carbohydrates. And we talked way back in our macromolecule conversation that carbohydrates are the first source of energy, but cells can also use lipids and proteins as possible sources of ATP. And so when we talk about making ATP, obviously the raw ingredients for that is coming from our food, which are cells, right? Either plant or animal cells, for us. But what we're not automatically thinking about then is, okay, how does that cell make those food molecules? How do we get the carbohydrates and the lipids and the proteins that ultimately we're eating to make our ATP energy? And so that is the connection that we want to make here. So remember, carbohydrates, lipids, and proteins are our sources of ATP, and we're getting this from our food. So if we're heterotrophs, meaning that we're eating things outside of ourselves, heterotrophs in the eukarya domain include protists and fungi and animals, right? We're getting this from our food. If you're a producer, if you're a photosynthetic protist or plant, then your ultimate source is the sunlight. So we're seeing here our aerobic respiration equation. So we're starting with glycolysis, where we're breaking down glucose to make pyruvate molecules. Notice that we have an intermediary molecule highlighted here, G3P. We'll see this again in our conversation of photosynthesis, so let's pay attention to it here. And then your pyruvate, remember, goes on to the mitochondria for pyruvate oxidation and citric acid cycle and electron transport chain. And we're getting ATP produced along the way at each of these steps. So clearly when we're eating carbohydrate, or if you're a producer and you're making your carbohydrate, we're going to be funneling that into this process that we've already seen. Where do lipids come into play? You can break down your lipids to make either G3P or acetate. So you can feed that into your reaction at either one of these places. And then your proteins can be broken down to make either pyruvate or acetate. And so we can stick it directly into that citric acid cycle. And so we'll see that our proteins can insert themselves at different points along this reaction. But here is the interesting thing. The arrows do not go in just the one direction. We can reverse these. So we can take energy and we can take different parts, different molecules here and go in the reverse. So you can start with pyruvate and make G3P and then make glucose and then use those glucose molecules to make other carbohydrates. Or you can take either the acetate or the G3P and use that to build your lipids. Or you can take your pyruvate or your acetate and use that to help make amino acids to build your proteins. So not only is the food broken down in order to make your ATP, but we can see that we can use these same raw materials in order to construct these macromolecules for whatever the cell needs. So if instead of needing ATP for the cell, for a plant cell, and we need glucose in order to make cellulose in order to make our cell wall, then we can go that direction with it, okay? And that's the end of chapter 6.